Good morning. Thank you very much for the opportunity to present this communication at the 6th edition of the Global Experts Meeting on Frontiers in Neurosciences, FIN-2026. My name is Rebeca Mira Sanchez from the Institute of Environmental Sciences and Neurodevelopment, ICMYN, and the University of Murcia, Spain. The title of this communication is Air Pollutants, Endocrine Disruption and Their Association with Neurodevelopmental Disorders. The aim of this presentation is to analyze an increasingly relevant issue in neuroscience and environmental health: the possible relationship between air pollutants, endocrine disruption, maternal and infant thyroid function, and neurodevelopmental disorders. The main idea is that air pollution should not be understood only as a respiratory or cardiovascular problem, but also as a possible environmental factor related to neurodevelopmental vulnerability, especially during pregnancy and early childhood. Neurodevelopmental disorders have traditionally been studied from a genetic, clinical, and neurological perspective. This perspective remains essential. However, in recent decades, there has been growing interest in environmental factors that may participate in the origin or modulation of these disorders. Today, we know that neurodevelopment does not depend only on genes. It also depends on the environment and on the moment when certain exposures occur. For this reason, there is increasing discussion about gene-environment interaction, epigenetics, environmental epidemiology, and developmental toxicology. The developing brain does not grow in isolation. It develops within a biological maternal family, social, and environmental context. Therefore, looking beyond genetics does not mean reducing the importance of genes. It means completing the picture. Air pollution has traditionally been considered a respiratory and cardiovascular problem, and it certainly is, but this view may be too limited. Recent scientific evidence suggests that air pollution may also be relevant to brain development, endocrine regulation, and certain behavioral outcomes. Air pollutants may include suspended particles, traffic-related pollutants, heavy metals, hydrocarbons, pesticides, and compounds capable of acting as endocrine disruptors. The question is not only what effect polluted air has on the lungs. The question is also whether these exposures may influence maternal physiology, the placenta, the endocrine function of the newborn, and ultimately neurodevelopmental trajectories. The timing of exposure is a key aspect. During pregnancy and early childhood, the brain is in a very rapid and highly sensitive phase of development. During these stages, essential processes take place, such as neuronal proliferation, neuronal migration, the formation of synaptic connections, myelination, and the functional organization of the nervous system. These processes require a stable biological environment. Hormones, inflammation, oxidative stress, and epigenetic regulation may influence brain development. For this reason, environmental exposure during pregnancy or during the first years of life may have a different importance from the same exposure occurring at later stages. In neurodevelopment, when may be as important as how much. The thyroid axis is one of the most important biological systems for early neurodevelopment. Thyroid hormones are essential for fetal and infant brain development. They participate in brain maturation, neuronal migration, synaptogenesis, myelination, and cognitive and behavioral development. During pregnancy, adequate maternal thyroid function is fundamental for the development of the fetal nervous system. In addition, even mild or subtle alterations in thyroid hormones may be relevant if they occur during critical windows of development. This point is especially important when we discuss iodine deficiency, environmental pollutants, or endocrine disruptors. Thyroid function is not a secondary aspect. It is a central component of early brain development. Some air pollutants may act as endocrine disruptors or interfere with hormonal signaling. This does not mean that all pollutants have the same effect or that all exposures produce the same consequences. However, the available evidence supports the hypothesis that certain environmental pollutants may alter endocrine systems, including the thyroid axis. Relevant exposures include suspended particles, traffic-related pollutants, heavy metals, hydrocarbons, pesticides, and other compounds with endocrine disrupting activity. These exposures have been associated with alterations in maternal and infant thyroid function, and if thyroid function is essential for brain development, then this endocrine pathway may be a plausible biological bridge between air pollution and neurodevelopment. Prenatal exposure and newborn thyroid function. In recent years, several epidemiological studies have associated maternal and infant exposure to air pollutants with alterations in maternal, neonatal, or infant thyroid function. One relevant study was carried out in California in a sample of 2,050 newborns. Prenatal exposure to airborne suspended particles was associated with an increase in neonatal TT4. This result must be interpreted with caution. It does not demonstrate that air pollution directly causes thyroid disease. Nor does it mean that prenatal exposure alone explains neurodevelopmental alteration. However, it does suggest that prenatal exposure to airborne particles may be associated with measurable changes in thyroid biomarkers at birth. And this is important because it connects three fundamental elements: prenatal exposure, air pollution, and neonatal thyroid function. In other words, we are not speaking only about an abstract hypothesis, but about a possible observable biological signal. Plausible biological pathways. The relationship between air pollution and neurodevelopment probably does not depend on a single mechanism. Several plausible biological pathways have been proposed. These include oxidative stress, neuroinflammation, endocrine disruption, alteration of the thyroid axis, epigenetic changes, and effects on brain development. These pathways should not be understood as isolated compartments. They may interact with one another. For example, pollutants may increase oxidative stress. Oxidative stress may promote inflammation. Inflammation may affect the placenta or fetal signals. And endocrine disruption may modify regulatory processes involved in development. Within this set of mechanisms, the thyroid pathway is especially relevant because thyroid hormones are necessary for brain maturation. The strength of the argument is not in saying that one single mechanism explains everything. The strength lies in the fact that several lines of evidence point in a biologically coherent direction: air pollution and brain structure function. The next level of evidence concerns brain structure and function during development. A 2025 systematic review analyzed exposure to air pollution and brain structure and function during development. This type of evidence is important because it goes beyond merely measuring environmental exposure. It asks whether that exposure may be associated with observable changes in the developing brain. Again, we must be cautious. Air pollution does not determine brain development by itself. Neurodevelopment is multifactorial. However, the available studies suggest associations between exposure to air pollution and changes in brain structure or function during development. This reinforces the need to include environmental pollution in the study of neurodevelopmental risk factors, PM2.5, and neurodevelopmental outcomes. Fine particles, especially PM2.5, are one of the most widely studied components of air pollution. In 2025, a systematic review analyzed prenatal exposure to PM2.5 and its chemical components and their effects on neurodevelopmental outcomes in neonates. The evidence collected in this systematic review indicates that prenatal exposure to PM2.5 is associated with an increased risk of autism spectrum disorder, ADHD, and impairments in motor and cognitive development, particularly in studies conducted in the United States and China. This is relevant because PM2.5 particles are not simply dust. They may carry different chemical components with different toxicological profiles. Prenatal exposure to PM2.5 deserves special attention because it occurs at a stage when the fetal brain is particularly vulnerable. We cannot reduce neurodevelopmental disorders to exposure to PM2.5. That would be far too simple. However, the evidence does support the need to study fine particles as possible contributors to early neurodevelopmental vulnerability. Autism and other neurodevelopmental disorders. The clinical question, therefore, is whether these environmental pollutants are associated with disorders such as autism spectrum disorder, ADHD, and other neurodevelopmental disorders. At the end of 2024, another systematic review examined the contribution of environmental pollutants to the risk of autism and other neurodevelopmental disorders. This included studies on nonlinear dose-response exposure to certain metals and ASD risk. The review concluded that, overall, studies indicate that exposure to several pollutants, including airborne fine particles such as PM10, during the first 3 years of life was significantly associated with the risk of neurodevelopmental disorders. This is an important point, but it is also a point where scientific caution is essential. Association does not mean direct deterministic causation. Autism and ADHD are complex and multifactorial conditions. Genetic susceptibility, prenatal factors, the early environment, inflammatory processes, endocrine mechanisms, and other biological and social factors may all be involved. Therefore, the message is not that air pollution alone explains these disorders. The correct message is that environmental pollutants may form part of the risk landscape and should not be ignored. What does the evidence suggest? So, what does the evidence suggest overall? First, there is growing concern about the relationship between environmental exposures and neurodevelopment. Second, there is biological plausibility. Air pollutants may be related to oxidative stress, neuroinflammation, endocrine disruption, thyroid alteration, and epigenetic changes. Third, the timing of exposure is key. Pregnancy and early childhood are critical windows of vulnerability. Fourth, the evidence must be interpreted with caution. Studies may differ in how they measure exposure, which outcomes they assess, when measurements are taken, which confounding factors are controlled for, and which population is analyzed. Therefore, the strongest conclusion is not a simple causal statement. The most rigorous conclusion is that the available evidence supports the hypothesis that air pollutants may contribute to neurodevelopmental disorders during critical windows of vulnerability in brain development, and that further research is needed to clarify mechanisms, windows of exposure, and preventive strategies. Prevention and public health. The public health implications are very important. If air pollution contributes even partially to neurodevelopmental risk, then reducing exposure during pregnancy and early childhood should be considered a preventive priority. This does not depend only on individual decisions. It also involves urban planning, reduction of traffic emissions, industrial regulation, environmental monitoring, maternal health programs, and pediatric prevention. The protection of neurodevelopment must include environmental health. Pregnancy and early childhood should be considered priority windows for reducing exposure to pollutants. This approach is not alarmist, it is preventive, and it is also consistent with the precautionary principle. When there is a plausible environmental risk affecting vulnerable populations, prevention should form part of the health and social response. Take-home messages. There are 3 main messages worth retaining. The first, air pollution should not be understood only as a respiratory or cardiovascular problem. It may also be relevant to neurodevelopment. The second, endocrine disruption and pathways related to thyroid function deserve special attention because thyroid hormones are essential for fetal and infant brain development. The third, prevention during pregnancy and early childhood should be considered a public health priority. The evidence is still developing and many questions remain open. However, the convergence between neuroscience, environmental toxicology, epidemiology, and endocrine biology makes this field an especially important area of research. As a final conclusion, air pollution should be considered a possible environmental risk factor for neurodevelopment. The available evidence suggests associations between air pollutants, endocrine disruption, alterations related to thyroid function, and possible adverse effects on neurodevelopment. These findings do not justify simplistic causal statements. However, they do support a clear preventive message. reducing exposure to environmental pollutants during pregnancy and early childhood may contribute to protecting neurodevelopment. Thank you very much for your attention.